So before we start the main topic of today, I'd like to just spend a few minutes introducing Singleron Biotechnologies. We are a single-cell sequencing company. We provide instruments, reagents, as well as services to support customers' end-to-end single-cell sequencing workflow. So one thing that's setting us apart for single-cell sequencing is we put a lot of emphasis to the steps even before single-cell sequencing. So that's tissue processing and sample handling. So here I would like to just share some quick data. As we know, the quality of single-cell suspension is essential in single-cell analysis. So that's why at Singleron, we come up with not only specialized protocols for different tissue and cell types, but we also have reagents and automation solutions that allow researchers to automatically perform single-cell dissociation as well as nuclei extraction so that they can have high-quality single-cell or single-nuclei suspension to get started for their single-cell workflow. So this also allows them to process multiple samples at the same time to cut down not only processing time, but also preserve cell viability and nuclei quality. So the topic today is neuroscience. So I would also like to share one of our previous example. This is from the University of Luxembourg by Professor Jens Schwanbaum. So he used human induced pluripotent stem cell to produce midbrain organoids that contain a wide range of cell types. And he used single nuclei sequencing to look into the performance and behavior of the different cells in these organoids, which captured some early phenotypes of Parkinson's disease. So if you are interested to learn more about his study, you can also check out the YouTube video after the webinar today, where he shared some of the details and insight from his research. So I highly recommend that. And so we, of course, are looking into more than just transcriptome. So at Singleron, we provide a wide range of products that add additional layer of data on top of RNA. So, for example, we have this immune profiling product that look at VDJ sequences, regardless of if you are only looking at the CDR3 region or if you are looking at the full-length VDJ sequences. We also have focus scopes that allow you to look at targeted sequences, including mutations, gene fusions, and even non-poly-A RNA sequences such as viral transcripts. We have other products that look at specialized results such as glycosylation, as well as RNA dynamics. That dives into not only how many copies of transcripts are present in the individual cells, but how fast are they produced or are they degraded, so that provides additional insights into your studies. So we also have sample multiplexing product that allow you to pull multiple samples into a single run to save you time and also reduce cost, especially if you are running large-scale single-cell experiments. The last but not the least, our latest product, MobiScope, look at full-length RNA sequencing, so that regardless of the relative location of your sequence of interest, no matter it's near the 3' or 5' end, you have a good chance of capture that using MobiScope. Yes, I guess that's enough about Singleron. So what if we can add even one more additional layer of information, which is the energy profile on the single cell level. So here I would like to pass the time to Yan, who is the managing director at Doppelganger, who will share some very interesting data that's a joint collaboration between Singleron and Doppelganger. So Yan, over to you, please.
Thanks a lot, Yingting. Thanks a lot for the introduction and the overview on the sequencing technology, which is really important for us because if you want to do single cell metabolism, then we obviously need good data, and the tissue dissociation is a very, very critical step. This is how we came across Singleron. So let me start with a question. And this is, if we could quantify the energy status of every single cell, what could we do? And there's a bunch of things we would be able to do if we were able to measure the energy production and the energy consumption in every single cell. First and foremost, we could do a functional stratification of seamless homogeneous cells. So cells that are able to produce enough ATP, and those that are maybe not in the position to produce enough ATP, we would be able to differentiate between that. We would be able to see metabolic zonations in tissue, and we could certainly augment histopathology. And more important, we would have a very, very functional and a very, very central marker for drug response and also drug resistance, because the energy metabolism obviously is a very central metabolism. So what I want to show you today is a technology enabling you to do a metabolic phenotyping from bulk samples, such as tissue biopsies, for example. You can do metabolic phenotyping on a single cell level and also on the spatial level. What we do is metabolic phenotyping. We're not doing metabolomics. So we're not measuring concentrations, we are measuring rates, very similar to the seahorse system that I will show you in a minute. This has nothing to do with gene set enrichment analysis, and this has nothing to do with AI. It's a bioanalytical, it's a measurement platform. And you might okay. And you might be familiar with a platform, an Agilent platform called Seahorse. And this platform basically quantifies two parameters, the OXPHOS rate and the oxygen consumption rate. And typically you can measure the basal rate, the max rate, and you can calculate then the metabolic reserve. We do a very similar thing using a very different method. And we typically look at ATP production rate and the oxygen consumption rate, exactly like the seahorse does. We also look at the respiration efficiency, and then going one step deeper, we can tell where this ATP is coming from. Is it mainly glucose driven? Is it lactate driven, so the carbohydrates, or is it the fatty acids? Is it branch chain amino acids, which are of minor importance, or is it maybe the ketone bodies driving this process? And moreover, we not only look at more parameters, but in contrast to seahorse, which requires cell suspensions, we can work with tissue, actually including FFPE tissue biopsies. We can work with single cells. And as I mentioned, we can also do spatial biology. We use the Visium or the Geomics platform for that. And all this was always cross-validated against the Seahorse platform. So here on the right side, you look at a brain organoid culture, which was treated with 3 different drugs. On the left side, you look at the seahorse data. On the right side, you look at our data, and the results are basically the same. In the next 20 minutes, I will explain the QSM technology, how it works. I would walk you through some key applications, and then I would summarize this neurology study that we did on Parkinson's patient biopsies that have been summarized in this application note. And we will make this application note available to you so you can read the results in pretty much detail. I'm going to highlight some of the services that we are providing in collaboration with Singleron, and then we leave some time for Q&A. So the QSM technology is a typical bioanalysis technology. We work with cells or tissue biopsies or organoids, be it 2D or 3D. If possible, we do an enzyme-plex assay using a proteomic platform. And here we quantify 518 enzymes plus the isoforms of these enzymes. If this is not possible, like in a single cell application, like I'm highlighting today, we work with an assay called enzyme-plex. It's a sequencing assay, and we use the RNA-seq data to convert into an in silico proteome, which is not exactly as good as the real proteome, but we're getting pretty close now. This abundance data is then imported into our platform, and the calibration curve, in contrast to a conventional calibration curve, has three functions built in. Number one, it's the network. So the calibration curve covers the entire central metabolism, the pathways that are catalyzed by these 518 enzymes. But we also take into account the regulation of each of these enzymes, interconversion rates, phosphorylation, and so on. And then the third layer is a systemic regulation. There are feedback loops in the system. For example, if I eat like 5 Snickers right now, my glucose metabolism will face feedback loops, and this is also considered in the platform. And again, the platform was validated against the Seahorse platform, as you can see here. And we also validated the turnover rates that we quantify against chemical and biochemical assays. Overall, we validated 177 parameters, and we're going to make this presentation available to you so you can also see the publication where the data has been published. And now we can work with basically every sample type. All these samples that have not been compatible to Seahorse, they fit into our platform. We can work with tissue, number one. And here I show an example from a tissue study which was done on a low-grade glioma, overall 49 subjects. And IDH1/2 mutant is a rather rare gain of function mutation, and these patients basically lose energy. And this study was done by Servier, a French drug maker, and they evaluated 2 drugs, ivosidenib and vorasidenib, and we measured the effect of the drug on the ATP production rate, on the oxygen consumption rate. And as you can see here, the vorasidenib was significantly more effective, bringing up the ATP production rate measured in micromole per gram per hour. And not a surprise that this is reflected also by the oxygen metabolism, because obviously ATP production and oxygen consumption typically run in parallel. So you could really see the beneficial effect in terms of efficacy of vorasidenib. Then going one step deeper, we were also able to elucidate that this effect was mainly coming from an increased fatty acid oxidation. In the meantime, this drug, vorasidenib, has been approved and is on the market. So you can work with tissue, something Seahorse can't really do. You can also work with single cell suspension. So here we look at a biopsy from hepatocellular carcinoma, a liver cancer, and we look at different cell types being part of the liver, and we look at the ATP production rate, and you see the Tregs here on average have an ATP production rate. First, you see that not all cells have the same ATP production rate. They differ in terms of their ability to produce ATP. And the Tregs on average have a higher ATP production rate, for example, compared to the mate cells. And again, we measure the basal rate, we measure the maximum rate, and then we can calculate the metabolic reserve. Now, interestingly, you can now define very specific subpopulations. And here, for example, when we look at the ATP produced from fatty acids, all the Tregs are high fatty acid consuming, high ATP producing cells. Whereas the Th1, Th17 subpopulation divides into high performers and low performers. So you can really see that a seemingly homogeneous population is very different in terms of the ability to produce ATP. And the third level, excuse me, is a spatial level. Here we use RNA sequencing data coming from the Visium platform. On the left, you see the H&E stain of a mouse brain biopsy. And now you can look at the glucose uptake rate, the oxygen consumption rate, the ATP production rate, and also the respiration efficiency. And surprisingly, the oxygen uptake is higher in the cortex and in the hippocampus. And not a surprise, but those areas which are more, which take up more glucose, also do more respiration. And it's not a surprise that those areas are also produce the most ATP. It's almost twice as high as compared to the low productive areas. And also we can see that the brain can tune the respiration efficiency, which is shown in graph E. So those are the three dimensions. And now moving towards neurology, moving towards single cell RNA sequencing, it was really important that we get a crisp separation. And this is really the dissociation is really key. Our platform can work with single cell data, also single nucleus data, and this is all summarized in the application note. Before we started the project, we had experience on a microelectrode array where we looked at neural cells. And MELAS is a disease where the neurons suffer from a reduced ability to generate ATP. And you can see here in the study, we were able to measure the glucose uptake rate and the ATP production rate. And second, we did a study on the single cell level, and we looked at Leigh disease, again a disease where the Leigh syndrome is categorized by mitochondria which are impaired, and we were able to measure again oxygen consumption rate and ATP production rate, glucose uptake rate, fatty acid uptake rate, and ketone body uptake rate. And you can really see how the Leigh organoids were affected by the disease. So we had confidence that we can work with tissue biopsies. We had confidence working with single cells on an organoid level. And then we moved to the Parkinson study together with our partner, Singleron. The study was conducted by Mainz University. You may know Mainz because it's the home of BioNTech, where the COVID vaccine was invented and manufactured. And the university clinic had access to 14 subjects. 9 were diagnosed with Parkinson and 5 non-Parkinson control subjects. The inclusion criteria were the diagnosis with movement disorders by a specialist, and the patient were also tested by a clinical neuropathologist to exclude cognitive impairment. And we got access to brain samples that were obtained during periprontal cortex surgery. And on average, these biopsies had size of 50 to 100 microgram. So very interesting study. Singleron did the single cell sequencing, and we excluded all the smaller populations. So at the end, we looked at a bit more than 83,000 cells, and we decided to look at 4 different cell types: the astrocytes, the oligodendrocytes precursor cells, and the microglia. And as you would imagine, there's different proportions of these cell types reflecting the situation in the brain. And now you can see some really interesting results here. So first of all, in the PD group, which are marked by PD after every cell population. First of all, you see, and we look at the ATP production rate, we see that the ATP production is elevated in every cell type. The astrocytes have a really significant, I mean, this is the brain, this is not an organ which is up and down regulated. Brain cells that have such a significant difference in ATP production rate, they are really affected by Parkinson. Same is true for the microglia, same is true for the oligodendrocytes precursor cells, same is true for the oligodendrocytes. But there's a second thing which is really important and which we saw sort of immediately. When a brain is constantly inflamed, and obviously microglia play an important role in this chronic inflammation, you see that some of the microglia are obviously stimulated, so they need to produce more ATP. I mean, inflammation is really energy consuming. And this is why many of the cells show an elevated ATP production rate. You see that the control has a very sharp band here. So the amount of ATP that microglia typically produce is very constant. But in the Parkinson microglia, we really see an elevated level. At the same time, we see many microglia showing a reduced level. And this is a phenomenon that we know from chronic inflammation. There's these mitochondria in the microglia, they undergo rapid aging as an effect of the constant inflammation. So for example, from longevity projects, we know that aging is very strongly correlated to constant elevated levels of mitochondrial activity. So here really, I think this is a very good marker showing the constant inflammation at the same time increased aging. There's one more thing. I mean, the ATP and the energy metabolism is only the first level. We can go one step deeper and we can look at the energy metabolism. And in dark blue, we see the carbohydrates, so the uptake of glucose and the uptake of lactate. In red, we see the ketone bodies, and we see that ketone bodies don't really play a significant role. The most important energy providing substrates are fatty acids, and a small portion, just 1 to 2%, is provided by branched chain amino acid. And what you can see here in the astrocytes, for example, is that, again, fatty acids play an important role. It's the outmost important substrate for providing ATP in the brain. But the carbohydrates also play a relevant role. And if you will, these astrocytes turn glycolytic. We see an increase by 3 percentage points. So a really significant increase in glucose and carbohydrates, in carbohydrate uptake rates when cells, as compared to Parkinson astrocytes. This is not the case in oligodendrocytes. So it's not that Parkinson converts all brain cells and causes kind of a glycolytic switch. This is not the case here. This is obviously a cell type specific effect, very similar to the cell type specific aging effect that we see in the microglia. So a lot of things that can be discovered, and you can access the full white paper through our website or through our partner Singleron. And to conclude this presentation, if you're interested in other disease areas like metabolic diseases, then on our website you will find quite a few posters that we did together with some of our clients. There's a poster on NASH together with Novartis. There's a poster together with Lundbeck on Parkinson's, and there's a poster on colitis together with AbbVie. The Enzymeplex products are available through us. We basically provide services, so people send us samples. We run the analysis, and these are the 9 service packages that we provide, focusing on neurometabolism, number 3, but also cardiometabolism. Triomics does proteomics and transcriptomics and metabolic phenotyping. The hepatic metabolism is a bit of a special metabolism. Then the general OXPHOS metabolism. We also do safety testing. I'm not addressing this today. We do single cell metabolism, immunometabolism, and cancer metabolism. And if you consider using or just trying our QSM platform, and you have samples, but you don't have data yet, then please contact Singleron, and I'm sure Yingting will be happy to provide contact details, because they also provide our services as an add-on to their services. If you have data ready already, then feel free to contact me simply through my LinkedIn profile, where you can book an appointment. We typically, for attendees of our webinars, we do the data QC free of charge, and through this we want to make sure that we can really work with your data, that we can really provide you with results. We do smaller pilot studies, we do full-blown clinical studies, as you have seen. And what we see increasingly over the last couple of years really is that researchers having limited access to samples, maybe they work on a rare disease and maybe they have 3 patients and they have generated a hypothesis. We can work with data that we download from a data repository, so we can get access to another, let's say, 37 patients, and then we have a second data set that we can use to verify these research hypotheses. On our website, you will also find a number of testimonials focusing on stem cells, cardiology, neurology, oncology, but also there's a bunch of other diseases and therapeutic areas like reproductive medicine. Just check out our website to see what is available. And with this, I would like to hand over back to Yingting, and I would like to open the Q&A session, please.
Yeah, thanks, Ian, for the presentation. So for the webinar participants, if you have any questions for Ian, you can use the Q&A function, which is at the bottom of your Zoom screen, and I will read out the question to him. If you can submit your questions now. Yeah, maybe in the meantime, Yan, I do have a question for you. So I think metabolism is often overlooked in research studies. So do you have some favorite research areas that you think metabolism will play an important role, but people often are not looking into that?
Yeah, so it's a very good point, Yingting. And I think what we really see is that over the last 20 years, a lot of metabolomics has been done, and we measure metabolites, we try to correlate that, but a metabolite concentration does not always really explain an effect. It's just we do correlation analysis, but correlation obviously is not implying causation. And what the field really is missing is functional marker where we really can pinpoint here is a limitation of a biological system, and this disease is, for example, caused by a lack of energy or by a lack of substrate or by an enrichment of certain substrates. And in cases where we really need functional information, this is oftentimes neurology, because neurology really depends on the energy metabolism. It's not so much the glucose metabolism or a detoxification metabolism. It's really, can these cells convert glucose fatty acids into ATP? But also cardiology. Obviously, a lot of cardiac diseases are rooted in the fact that cells have a problem in generating enough ATP. The other way around, immunology. We see a bunch of studies where in chronic inflammation, cells are constantly stimulated. And obviously, inflammation takes a lot of energy, but this causes difficulties for the cells down the road. So I would say oncology, I should have missed that. Oncology is really important because obviously we see that in the tumor microenvironment, we all know the Warburg effect. The energy metabolism plays a really central role. So I would say it's oncology, it's neurology, it's immunology, and it's some of the metabolic diseases really that are most important. And
Yes,
with
thanks.
this combined offer, I think customers now really have the opportunity to look at gene expression, and this is great to generate hypotheses. And then you can combine this with a very holistic metabolic view in order to verify these hypotheses.
Yeah, that makes a lot of sense. We have a question on, it seems that the analysis is very sensitive. So what's the requirement in terms of control samples and also sample quality?
Yeah, very good. I mean, obviously, let me start with sample quality. Obviously is this sort of crab in, crab out problem. Yingting, you know this from single cell RNA sequencing. If these samples have undergone some degradation, then obviously we see this. Pre-analytics is really key. So we highly encourage customers using PaxGene or other platforms to preserve the RNA profile really. But other than that, if RNA degradation and RNase activity have been stopped, you can work with every sample type, being it a cell suspension or a tissue sample. Even FFPE blocks are accessible now. The amount of RNA which is needed, I would guess, Yingting, this is probably more on your end, because you are really the expert on RNA sequencing, providing great data on samples. So what would be your recommendation?
I guess for the single cell and single nuclei analysis side, it depends a little bit from tissue to tissue. So we typically recommend if it's in terms of viability. So feel free to get in touch with us if you have a specific tissue in mind. But it can be anything from a few hundred thousand of cells, or we have even processed lower number of cell input. That's also possible. Yeah.
And I think this is really where Singleron's expertise is in this field. We tried our platform in combination with single cell data coming from Singleron and single nuclei data from Singleron. Both works. But the project managers at Singleron really make sure that you pick the appropriate sequencing protocol for a specific sample type, I think.
Yeah, so Yang, we also have another question in terms of what level of spatial resolution can QSM achieve when you overlay that with transcriptomics?
Yeah, so basically, as we work with transcriptomic data at this point, we are using the platforms like the Geomics and also the 10x Visium platform, we basically rely on the resolution that these platforms allow us to work at. And we work with those protocols where the spatial analysis is combined with next-gen sequencing. So again, it's kind of an add-on analysis using an existing platform. So the answer in short is it's the same resolution because we start with the same data. We do a very different analysis. It has nothing to do with gene set enrichment or so, but it's the same data we start with. It's just using the same data, looking at it from a very different angle, using the same data.
Yes, thanks. I hope that answers the question. We have another one that's more on the practical side. I think for the QSM platform, what's the cost and turnaround time?
Let me start with the turnaround time. So let's assume Singleron has completed the sequencing already. Then, and it depends a little bit on the complexity. In theory, it takes about 3 weeks. In reality, we often see that after a first look at the data, people have additional questions, and we can often address these questions. So let's say we have a preliminary analysis. We look together with the customer, we look at the data, and then we do a second round of digging deeper into, for example, substrates. And then it takes approximately 5 weeks. And the costs are somewhere between 3,000 and I would say 15,000. Really depends on the number of samples and depends on the complexity and also on the number of comparisons that we have within a project, really. Yeah, I should mention that for single cell analysis, our preferred partner is really Singleron because we have a workflow up and running where we know that it works and people know each other. So it's really a very smooth workflow that we have established here.
Yeah, thanks so much, Yan, for this collaboration. I think that's pretty much all the questions we have. Thanks for all the audience as well for participating actively in this webinar. We're really happy to work with Doppelganger with your single-cell metabolics analysis needs. And after the webinar, we will follow up and share with you both the presentation slides for today as well as the app note, as Yan has mentioned. So feel free to reach out directly to him if you have any questions regarding the QSM platform, and I'm sure he will be more than happy to answer that. Yeah, so with that, thanks everyone for being here today. Thanks, Yan, for joining us for this very insightful discussion. And we look forward to seeing everyone at our next event.
Perfect. Thanks a lot. See you soon. Have a great day.
Bye.